Okay, welcome back, everybody. How we doing? Chapter 16, DNA structure and replication. Now, before we get started, I want to share something with you. One of my favorite authors is Sam Keen, and he's the author of maybe 4 or 5 science books right now. He's a great storyteller. And this book is called The Violinist's Thumb, and it's the history of genetics. It's fascinating. If I could teach a class that I would most want to teach, it would be the history of biology, because the ideas that have been shared and the people who have corresponded with each other to give us our scientific discoveries fascinates me. It gets right to the core. Awesome book. I'm going to leave a copy of this on audio, so the MP3s on Classroom, so you can listen to it anytime you want. I highly suggest it. Chapter about Mendel is awesome. So let's start with what we have to know. The AP, this AP structure that we're going to have to use, is very broad when it comes to genetics. I had to cut out the first 2 pieces, cut out and put onto the slideshow so that we would understand what it is we're going to look at. So we're doing DNA and RNA structure, and we're going to do replication. This is sort of long, so I had to bust it up into 2 videos. In the first video, I'm going to do a little history about the discoveries that led to the structure of DNA. And this isn't for my own sake. This is something for the AP course. They want you to know about these scientists. And then in the second part of that video, we're going to look at the structure of DNA overall. Second video, we'll tackle the machinery and mechanisms of DNA replication. So my targets, I want you to be able to explain and identify important experiments that gave us the understanding that 1, DNA is the heritable material, and 2, gave us the idea or the concept of the structure of DNA as we now know it today. You're going to understand the difference between DNA and RNA a little bit better at the molecular level. And then, as I said at that second video, we'll tackle replication, but that's for a little later. What we're going to talk about was mostly discovered on the type of the lab you see on your left. The lab on the right, man, that is a modern lab. 50% of what we talk about in this unit was known before we got any really serious molecular genetics and technology involved. Fruit flies, mice, viruses, bacteria. This was the go-to for decades, and it got us to understand the molecular structure without all the weird technical stuff that we have today. Here's the list of the big experiments that you're going to have to know. And I would suggest going through this video a couple times, but they tell a story. One builds on the other. All right. And the big one for getting to DNA structure is the one that's highlighted inside the circle, like 50 through 58. That was the golden era of DNA structure. Right. So we've known about the structure of DNA since the mid-50s, since the Korean War, for crying out loud. So let's start with the first one. This is called Griffith's experiment, and he did it on mice. So what you start with is you have some living cells that are S cells. S stands for smooth. They have a sort of a gel capsule around them. And then R stands for the rough. They lack a capsule around them. One is virulent, one is not. So when you take these S bacteria and you inject them into a mouse, the mouse will die. That's because this is called a plasmodium. This gel on the outside does not allow the immune system to detect that it's there, and it's allowed to go about its business, and then it kills its host. Tuberculosis is like this. On this other side, it's got plenty of little proteins on the surface that the immune system picks up on, so it's running around naked in a sense, and it gets destroyed, and then you end up with a healthy mouse. So this is what Griffith did. He said, what if I boil, heat kill all of these S bacteria, and I inject those S bacteria, remember the ones that kill, into a mouse? What's it going to do? Mouse lives. Okay, so we now know that if you kill the bacteria, it can't kill the host. Now, he took these dead bacteria and he put them in with the non-virulent R strains, mixed them together, let them grow together. And when he injected that mixture into the mouse, 2 crazy things happened. One, this non-virulent bacteria that he supposed wasn't going to harm the mouse killed the mouse. Surprise number one. In surprise number two, when he isolated the bacteria, they weren't the round bacteria anymore. They were the smooth bacteria with the capsule on the outside. He gave this phenomenon the phrase transformation because the bacteria that were once round and non-virulent had been transformed into a virulent form. So these are the two discoveries that come out of it. There is a heritable molecule that we now know of. Didn't know it was DNA yet, had a hunch. And then it was also capable of giving or conferring those qualities onto the next generation or onto something else. That was, I mean, think about the phenomenon that they discovered right before the stock market crashed. They discovered that there's a molecule that when you give it to something, it will, the properties that you gave it will take over. That's, I mean, today we're like, okay, 1928, that's a pretty big deal. So that's Griffith's experiment. A few years later, 15, 17 years later, you have the Avery experiment. And in the Avery experiment, he did the same thing as Griffith, but he tried something with a little bit of a twist. He kills the bacteria, mixes them with the R again, but this time he uses stuff that breaks down RNA and then use those into the mice. He uses proteins, puts those into the mice, puts in something that breaks down DNA, puts those into the mice, right? So he breaks down all of the major organic compounds in identifying separate experiments. And in each of these, this one, this one, this one, and this one, all of them cause the mice to die except the one that had the DNA of the S strain destroyed. That's brilliant. 1942, we're getting ready to go into World War II. That's where this experiment was happening. That was a really nice experiment. That's an elegant experiment. Has a lot of different variables and you're isolating each variable. And now we know that once you remove the DNA, you don't confer those qualities anymore. Kudos, Avery. Good experiment, dude. All right. So this brings out that there is a heritable molecule and it's going to be DNA. And it expands upon Griffith's previous experiments. Okay. Now we start getting into the meat. We now know it's DNA. We now know that there, first of all, there's two camps. There's the protein camp and there's the DNA camp, and they're warring all the time. People are really behind it because if you think about it, there's so many different kinds of proteins, and there's only this jelly stuff inside the nucleus called DNA. DNA wasn't anything. It was just DNA, but proteins are everywhere. So proteins have to be the one that's passed on. Okay. Erwin Chargaff was doing some experiments on the side. He was a little weird. He was into isolating all the chemicals that lived in organisms. Let me rephrase that. He was into isolating all the chemicals that made up living organisms. What he found was that adenines and thymines, I know it doesn't look right here, but it does so down here, were often in very similar proportions in an organism. And that went for guanine and cytosine as well. Not only that, but it was also species specific. So if we talked about E. coli had a proportion of A to T and C to G that was different from what a chimpanzee had for the proportion of A to T and C to G. Maybe the bacteria had 20% and 20% AT, where the chimpanzee had 25%, right? So that's that, and it was species specific. He found this relatively the same. Now his numbers sometimes get poked at a little bit, but we're talking about 1953 with that ancient equipment I was showing you. He was still even able to come as close as he was is remarkable. But now in modern terms, we're able to nail it right down. And those proportions have really narrowed their standard deviation. They're very tight numbers now against each other. So what did Erwin show us? He said that A's and T's are in proportion and C's and G's are in proportion. And it was species specific. Excuse me, just had dinner. Species specific as well. Awesome experiment. The Hershey-Chase experiment is absolutely awesome. I just said that there was a protein camp and that there was a DNA camp. Each of them is trying to find the nail in the coffin, right? Which one of us is really going to show that it's our molecule that's the heritable molecule? So check this experiment out. We start with viruses. And viruses, if you don't know much about them yet, they're called phages, first of all. Bacteria have their own types of viruses, and they're called bacteriophages. Oops. Bacteriophages have an outside protein coat, and then the DNA resides inside the protein coat. When the virus replicates itself, it squats on a bacteria and it squirts its DNA into the host, and then the protein coat falls away. All right. So what they did was they said, all right, let's start with protein. And what are things that make up protein? There's carbon, there's hydrogen, there's oxygen, there's nitrogen, and there's sulfur. We can count on all of those to be in proteins. Well, what about DNA? Nucleic acids have carbon and hydrogen and oxygen and nitrogen, but they also have phosphorus. Well, they looked at those elements, sulfur and phosphorus, and knowing that there's no sulfur ever in DNA and there's no phosphorus ever in proteins, they used these and found isotopes of them and radio-labeled them. What do I mean by that? You put these organisms together and they need nutrients to grow. So some of those nutrients that you give them have isotopes of either phosphorus or sulfur. And they will incorporate that isotope into their molecules. Now that it's radioactive, you can trace it throughout its life cycle and beyond. Okay, so this is what they do. They take the bacteria and they allow them to infiltrate the bacteria in both cases. These are down here is radio-labeled DNA, down here is radio-labeled proteins. And then they put them into a blender, break them up so that they can get to the parts that matter. And then they put them into a centrifuge and spin them down. And this is what they found. That after the experiment was over, there was no radioactive protein inside the bacteria, meaning that all of the radio-labeled protein had been washed away. There was no transfer of heritable material from one generation to the next. However, generation after generation after generation, there was radio-labeled DNA in all the bacteria. And then the bacteria reproduces, and then there's more radio-labeled still down the road. 10 generations later, they're still following the radio-labeled DNA. What does that mean? It means that the instructions are DNA. Absolutely brilliant experiment. DNA is without a doubt the molecule of heredity. That's what this experiment showed. It showed that DNA was the only molecule that was passed from one generation to the next. Proteins are shed away. All right, man, pretty good. All right, so we're getting towards the end now. On my left, I have Linus Pauling. You will have likely someone who's a Linus Pauling in the schools that you go to. Linus oversees all of the grad work. He sees all the doctorates. So he had a couple really talented students working for him. The top one is Rosalind Franklin, and those two gentlemen below her are James Watson on the left and Francis Crick on the right. Linus Pauling wanted to discover the structure of DNA. Now the hunt's on. So it's almost, he wants to do it. He has these people working for him, but don't have no cloud of, what's the word I'm looking for? He wants it. He doesn't care what they want. He wants it, and he's using them. Okay. So what's Rosalind's contribution? Rosalind was an expert at X-ray crystallography. She could take molecules underneath this machine and she could get them to stretch themselves out, and then she would hit them with X-ray, and as the X-rays passed through the molecule and around the molecule, they would leave shadows. What you're seeing on the shadow is a crisscross of the DNA molecule as it goes by. This showed that it was double helixed. Her work ended up getting stolen, and that's where number 2 comes from. Women in science will always be robbed of the glory that they deserve. There is story after story after story of a woman either knowing way ahead of time what the answer was and nobody listened to her, Barbara McClintock, or they do all the hard work and they never get recognized. Rosalind Franklin, who was I thinking of? Madame Curie. And I'm sorry, I dwell on this a little too long every year, but it really frustrates me. I'll tell you more about her in a second, and I hope it frustrates you. Linus Pauling, in an effort to spur the gentleman on a little bit, actually shared some of Rosalind's work with them, but not vice versa. He didn't share any of their work with her. Well, this is what you get when you have guys who are really good at reading papers and really good at reading other people's work, but are really bad at doing experiments on their own. They were able, based on everybody else's work, Hershey and Chase, Chargaff, Franklin, some other experiments, they were able to piece together and know the structure of DNA before anybody else. To start with, it was already known what the diameter of the molecule should be if it was a double helix. If you took 2 purines like A and G down here and you put them in, there has to be space for the hydrogen bonding of the bases to go. That space put the edges of the nitrogen bases outside of that perimeter. If you just use pyrimidines, right, T's and C's, it didn't leave enough space. You were lacking. But if you, on one side you used a purine, and on the other side you used a pyrimidine, and you took into account the amount of bonding that they would need, they fit perfectly. Couple that with what Chargaff was doing, A's and T's, C's and G's, and you find out that a purine will always go with a pyrimidine, where a C and a G will go together too. Now this shows the structure of DNA, but you can win a Nobel Prize without doing a single experiment. No experiments were done. Everything previous to this said experiment. These guys did just a discovery. They won the Nobel Prize along with Linus Pauling because of his influence to the gentleman, but Rosalind Franklin didn't. Now, it's not because Rosalind wasn't appreciated. She wasn't mentioned for sure, but she ended up dying of cancer brought on by the X-rays because nobody knew the dangers of X-rays in those days before the Nobel Prize was awarded. It was a horrible story. And it sucks. Later on, here they are again with their other lab partners. Later on, the structure of RNA is discovered. So let's just focus on that for a second. DNA is a double helix, and RNA is single-stranded. Where DNA uses A's and T's and C's and G's, adenine uses 3 of the 4, but it substitutes thymine for uracil. Uracil is also a pyrimidine, just like cytosine and thymine, but it's sort of a chemical cousin. It's not nearly as stable. There is a lot of speculation as to why RNA uses uracil. There really isn't anything definitive. Maybe that's going to be your research project when you guys go out into the biochemical world. RNA is thought to be the original molecule for heredity, not DNA, so because it's so much more simple. And maybe it's just a carryover from the old sense of our molecular structure of RNA. I don't know. It is what it is. A couple other differences about it is that DNA, let's start over here with RNA. At the 2 position, there's this OH group, hydroxyl group. That O is missing on the DNA side. Therefore, DNA, by missing an oxygen, has been deoxygenated, and that's why it's called deoxyribose. This is a ribose sugar. Deoxyribonucleic acid, whereas RNA is just called ribonucleic acid because the OH is there, and that's where it gets its name, DNA, deoxyribonucleic acid, ribonucleic acid. Okay. Those are the big differences between DNA and RNA, and because it's single-stranded, it's going to be able to take a lot of different shapes. RNA is going to take the shape of messenger RNA. It's going to take the shape of tRNA. It's going to hold an amino acid up here. It's going to have 3 anticodons on the bottom. It's going to help hold the shapes of ribosomes together. There's rRNA, which is sort of like a lasso that goes all the way around these proteins and holds the structure together. That's a ribosome, and that's actually where mRNA passes through. So there's RNA helping RNA do its job. Crazy. Okay. Difference between RNA and DNA. Now, the anti-parallel molecule. When Watson and Crick built the structure of DNA, one thing that they found, let's just leave it there, was that DNA is it goes in 2 different directions. If we start, I'm just going to draw it up here. If we start with the 5th carbon, and the 5th carbon goes down to the 4th carbon, which goes down to the 3rd, which goes over to the 2nd, which goes over to the 1st. And then off the 1st comes a nitrogen base. And up here is where the phosphate group is. And then 3 bonds, hopefully, to the next phosphate group, which then goes down to the next 5th group, which goes down to the next 4. You can see where I'm going with that, right? Well, this 5 to 3 pattern goes in one way. We talked about this in the beginning of the year. A to T, C to G, you can only add to a 3. So DNA gets built in this direction, right? But it's a molecule that has dimension to it. If you shake your right hand with someone and they use their right hand too, they're opposite of you, right? So the hands go together. But if they were standing on the same side as you and you tried to shake hands with both hands out, you'd end up with one hand hitting the back of the other hand. They wouldn't marry together. And that's sort of the way that DNA works. The right hand goes one way and the other right hand has to go the other way. So if this goes 5 to 3 in this direction, this other one has to go in the opposite direction. It's called anti-parallel. They're parallel, but they're going in opposite directions. And you can see it in this molecule here. There's the 5 prime, there's the 3 prime, they're going in this direction. There's the 3 prime, there's the 5 prime, they're going in this direction. Okay. Anti-parallel, 5 to 3. Okay. Now, one more thing to bring up. I said it a second ago that there's hydrogen bonds holding the helix together. If this were a ladder, right, and these are the sides that you put your hands on, the rungs that you step on for your feet are where the bases would be. The A's and the T's only require 2 hydrogen bonds to hold them together, whereas the C's and the G's require 3. This was an interesting discovery because where genes start for reading later on, there's a real long stretch of A's and T's before the gene starts so that the DNA can get apart. One more hydrogen bond doesn't sound like a lot, but if there's a lot of little bases in a row, you want to make as little energy use as possible to get into that gene. So breaking 2 hydrogen bonds to open up the gene is easier than breaking 3 hydrogen bonds. So there's loads of little A's and T's and spaces where the DNA has to open up to be read. Natural selection at its best. All right, so let's go back. Let's choose 3 experiments and describe the experiment and what it demonstrated. Make sure you get the names right too. Pause the video. Don't go back on your notes. Actually physically think about this. I'll wait. Pause the video. Okay. I'm glad that you did. Here we go. Here are the list of the experiments I left off the Messelson stall experiment because that has to go next after we talk about replication. But Griffith, right, that was the bacteria that transformed into virulent bacteria and were able to kill mice. Avery did it by eliminating different compounds that were found in different bacteria. If he'd got rid of the DNA from the virulent bacteria, it didn't make the other bacteria virulent either. Chargaff was the guy who discovered that A's and T's go together, C's and G's go together. Hershey and Chase are the ones that did the virus bacteria experiments. They used the tracers. They traced sulfur and they traced phosphorus. Phosphorus is the only one that's in DNA, and phosphorus was the one that was found generation after generation after generation, proving that DNA is the molecule of hereditary molecule. Rosalind Franklin, she was the one for X-ray crystallography, showed that it was a double helix, got all her work stolen, no credit. Watson and Crick, they discovered the final structure of DNA. DNA is an anti-parallel molecule. It runs in a 5 to 3 direction. A's and T's go together, C's and G's go together. You have a purine, like an adenine or a guanine. They go to a pyrimidine, cytosine or a guanine. If you remember, cytosine or a thymine. Come on, Mabel. Pyramids cut with pure agony, right? Pyramids, the pyrimidines cut, C-U-T, with pure, that's the purines, agony, A-G. Helps you remember which ones are which. All right, so that's going to end it for this video. If you need to watch it again, I would. And I will see you guys in a little bit.